Hi, my name is Autumn Vance,
And my name is Austelle Arsenault.
and today we will be talking about the auspicious genome of phage Austelle, a cluster B4 mycobacteriophage which was discovered fall 2021 at Merrimack College.
We are the students who discovered Austelle. Our phage was discovered in the mulch bed soil beside Mendel Pond, which is a man-made pond that is directly outside of Palmisano Hall at Merrimack College.
What you can see here on this petri dish containing the host, we have spotted Austelle on the right-hand side in comparison to two other phages discovered at Merrimack College, Hopi and Leo. In this panel, you can see our small plaques that are turbid, indicating a lysogenic lifestyle.
After we purified the phage, we did a DNA analysis. What you can see in comparison to one of our controls, phage lambda in lane 2, Austelle's DNA is of a similar high molecular weight, indicating an intact, high-quality genome. The concentration was 40.27 nanograms per microliter.
Hi everyone, my name is Michaela Richard and I worked on the Austelle annotation for genome for spring of 2022. I'm going to talk about some interesting features of Austelle's genome. This is an overview of Austelle's genome. This diagram is a famirator comparison of Austelle located in the middle, with the closest relatives apex on the top and prints on the bottom. Austelle has a typical B4 cluster genome organization with 96 predicted gene products and a total of 71,799 base pairs. Consistent with other cluster B phage, we noted Austelle's plaque morphology was hazy. However, Austelle's genome lacks an integrase gene, as do other cluster B phage. Similar to many other cluster B phages, Austelle's genome contains a reverse orientation gene, GP8, with homology to RUV-C-like resolvase, flanked by reverse orientation genes of unknown function, which are in turn flanked by terminase and portal protein. GP12 includes a predicted H&H endonuclease, which I will discuss further in this talk. The proximal half of the genome is mainly comprised of genes in the forward orientation. The distal half of the genome alternates between segments of forward orientation and reverse orientation genes, with the distal portion of the genome is comprised of short genes in reverse orientation with no known function. Other features consistent with cluster B genomic organization is a long tape measure gene GP26 and the presence of the gene encoding DNA polymerase 1 in GP54. Now I'm going to discuss a few features of Austelle's genome that we found particularly interesting. One feature of Austelle's genome that is typical of cluster B4 is the presence of a reverse orientation RUV-C resolvase, encoded by GP7, indicated in the purple here. RUV-C resolvases can resolve holiday junctions, suggesting this gene may enable phage-mediated recombination. One aspect of Austelle's genome that is atypical of B4 cluster genomes is GP12. A predicted reverse orientation H&H homing endonuclease flanked by a major capsid protease and a major capsid hexamer protein. The only other cluster B4 genomes containing an H&H endonuclease gene are Apex and Jamal. Recently published work shows that a Hafnia phage with variable plaque morphology produces large plaques with variants that retain the H&H endonuclease genes, whereas variants extracted from small plaques lack the H&H endonuclease. This suggests a hypothesis in which H&H endonuclease insertion within B4 phage genomes may influence plaque morphology. It is unknown whether this is the case for B4 phage like Austelle, but is a question for further research. In conclusion, Austelle is a phage-infecting Mycobacterium smegmatis isolated by the Merrimack College C phages team. Austelle begins to cluster B4 and has a genome of 71,799 base pairs and 96 predicted gene products. Its closest homologs are Apex, Prince, Frederick, and Roger that. Austelle's genome contains a RUV-C-like resolvase and H&H homing endonuclease, Further analysis may reveal information on how similar phages evolve when competing under stressful environmental conditions.